Hello everyone, welcome back. So in this session we will talk about the transcription in the prokaryotes. Everybody knows that what is the transcription is. It is the transfer of information from DNA to mRNA. So here we know that mRNA is being synthesized inside the DNA strand. So when the DNA is synthesizing an mRNA, it is following some rules and regulations. That means one strand is acting as a template strand, another strand is acting as a coding strand also, right? And here we see the promoter regions and also terminator regions, and also we see these structural genes also. Here to identify that certain strands, here DNA is the one which is separated like this. For our convenient we will do like this, and here it is the one which is having a third primer to fifth primer direction, and also it is having fifth primer direction to third primer direction. Now we know that these are the polarities also, we can say. So here it is the one which is having a polarity like this, and here also it is having a polarity like this. Now when we are talking, we know that there is a signals giving, like we can call it as a this as a tata also, right? So here it is also called it as a pre-now box also, because it was been having a base pairs like 10 base pairs also, which is been identified by this. Now we know that to proceed the reaction, means this process of transcription in the prokaryotes, we know that DNA dependent and also that is RNA polymerase is an enzyme which is coming and attaching. But what happened here is, to come and contact with the this enzyme to this strand Pribnow box and this starting point, it needs support. That support was given by the sigma factor. So this process of stigma factor attaching to the DNA polymerase like RNA polymerase enzyme, we call this process as an initiation process also, the first step in the transcription in the prokaryotes. Now these two of them will enter and will bind up here, right? So when they bind up here, automatically the direction will be started, and it is also having a rule of following that it can add up these strands fifth primer to third primer direction. Now when the T is present, automatically A is adding, and A is there, uracil is there, next T is there, adenine is there, A is there, again we, so like this continuous one strand will be formed of here. Like this is an example to tell you, right? This is in the further strands it will be following these directions. That means it is following the rule, that is fifth primer to third primer direction. Later on, then initiation after, we call this as a process called as an elongation factor. Once the mRNA strand is being formed inside the DNA strand, and it is the one which is coming out of the DNA strand. Now this is an mRNA strand which is having a polarity of the fifth primer direction to third primer direction. Now after that, the termination, that means the DNA polymerase DNA dependent RNA polymerase is the one which has to come out of the DNA strand, so that there will be not any bubble like structure in this DNA strand. To form this, there requires the one factor, call it as an rho factor. Rho factor is the one which is entering on, which is informing to the that RNA polymerase just to come out of the DNA strand. So RNA polymerase and rho factors, all of them are coming out of this. Now DNA strand will be back to their position, and this process where rho factor is involving, we call this as a termination process. Now the perfectly already mRNA strand is the one which is moving towards to synthesize in the proteins. The interesting point to note it down is, in the prokaryotes, this, for example, this is in bacteria. Inside the DNA strand is the one which has given information, and mRNA has been properly framed out. Before that mRNA comes out of the DNA strand, still it is in the middle only, it will be coming in contact with the ribosomes, and the process of translation will start off. So like this the proteins will be synthesized. So this is about the protein synthesis, that means process of transcription in the prokaryote students. I hope you understood better. Thank you.